My background is as a clinical biochemist working within the NHS and the independent sector. So I'm going to try and take some of what Rachel's been talking about and try and support you guys into gaining some information about mitochondrial dysfunction. Firstly, I want to make a distinction. I think Rachel has covered part of this, but there is a clear distinction between what we see regularly in patients, clients, in dysfunction, mitochondrial dysfunction and disease. But there's a common denominator throughout, which can overlap. So it's important to bear in mind that most people won't come across mitochondrial diseases, genetic diseases, as Rachel has mentioned. But there are cases of late developments. Now, what we tend to see overall, in many cases, is things like toxic exposure, environmental toxins, herbicides, pesticides, personal products. And there are quite a lot of biolab tests to support treatment function. So this is really what I'm going to talk about. I'm going to try and relate some of the actual tests that we can actually do within the laboratory to give you indication of what's going on at a cellular level. So let's talk about, just to mention that genetic defects such as dysregulation and succinate synthesis, MELAS, which is mitochondrial encephalopathy, lactic acidosis and stroke-like episodes. And there has been a case of a 60-year-old presenting with MELAS. So mitochondrial diseases you may come across, but they will be rare. Most of what we're going to be talking about will be dysfunction due to various other factors. Now, there's an important thing to mention is that we don't have in the laboratory routinely methods that will directly give you information about mitochondrial function. Majority of tests will be indirect, and I'll come on to some of those in terms of nutrients, etc. later. So there are comprehensive profiles that we actually do, which will give you some indication, and there's 1 or 2 markers within each of those profiles, and I'll just go through them briefly. So organic acid test is one test that can actually give you a metabolic snapshot of many actual disturbances, dysfunctions going on. But there is 1 or 2 markers within that organic acid that will be able to give you an indication of mitochondrial health. Succinic acid is an excellent marker of mitochondrial dysfunction because it's actually common in both the citric acid cycle and the electron transport chain. And you'll see that it's marker 24 on the profile actually, but I regularly see this when I'm trying to support interpretation of these tests. And I've probably spoken to quite a few of you over the last 2 years since I've been with Biolab. But the succinic acid marker is a marker that comes up with chemical exposure, and it's elevated with chemical exposures. Mycotoxins is another major issue, and probably around 30% of patients you see will have difficulty with mycotoxins, detoxing. Some people, even in the same household, exposure to water damaged buildings one person may have a real struggle detoxing and be really quite sick, and the wife, child or whatever may actually be fine. So there are variations within that. The GPL tox has got another marker, triglycerol glycine, and it's an intermediate metabolite and the catabolism of isoleucine. It's a marker for mitochondrial disorders resulting from mutations of mitochondrial DNA, and these can manifest from exposure to toxic chemicals, infections, inflammation and nutrient deficiencies. So it's really quite a good marker. And when you actually see the succinic acid marker on the organic acid test elevated, it's sometimes worthwhile actually going on to do a GPL tox. You can sometimes it's worthwhile doing all three, but certainly worthwhile once you've got a basic metabolic snapshot using the organic acid test to follow up if any of these other markers come. The last one that we actually do really quite well is microbiome assessment. There's quite a few different tests that we can use to give you an overall indication of microbiome, and the markers that come up there are absorption, digestion markers, and inflammatory markers as well. So those are overall biolab profiles that are really quite the number of requests we get are really quite high for these because they do inform of a great deal of information. There's two aspects that I'm really going to focus on. One is actually monitoring of some of the supplements that that we can support, give patients, clients to support, and two is certain vitamins and minerals that we can actually determine. Sorry, there's just got a battery warning low. Yeah. Sure. Anyway, I'll go on. Yes, diet, a huge role, because as you're going to find today, and you've already some of the aspects of how diet has an impact, so we've been discussed by Rachel. There is a mitochondrial theory of aging, as many people will come across. And dietary factors have a huge role to play in this. So okay. So there's a long list of nutrients basically that are really required and essential for mitochondrial health and mitochondrial function. Many of which we measure on a routine basis, and we can do these within individual tests, or we can actually, there are several profiles that will incorporate a small number of them. So from the TCA cycle, the important ones you can actually see, we'll go through the list. Thiamine, many of the B vitamins, obviously. Niacin, vitamin B3, really essential. And I'm sure that Dr. Maher will talk about that with CFS later. Iron, sulfur, magnesium, manganese, cysteine, and alpha-lipoic acid. Nutrients required in elements of TCAN, the electron transport chain, zinc, riboflavin, pyridoxine, B6, iron, and copper. Synthesis of L-carnitine and pyruvate dehydrogenase. Pyruvate dehydrogenase is actually the rate-limiting enzyme for aerobic respiration, and so that's really important. So you can actually exercise and get most of your energy initially from the glycolysis, so the anaerobic part of the pathway, and then you'll get it all stacked up. And pyruvate dehydrogenase is a rate-limiting enzyme for going into the aerobic side. So it's really a key enzyme, and these nutrients are essential. Electron transport chain, riboflavin, iron, sulfur, copper, and coenzyme Q10. So when I said the diet was really important, this is a really schematic of what essentially happens when macronutrients taken in. And in terms of sugar, so today's diet, as everyone will probably recognize, is that a large part of today's diet is processed foods, which generally have a really high level of carbohydrate. Carbohydrate is not a clean fuel in terms of mitochondrial health. There's a huge amount of free radical production, reactive oxygen species, which can damage the mitochondria. So this is really, and then you actually get the byproducts build up and cause actually cellular and mitochondrial DNA damage. The macronutrients, fat is actually much more clean burning. It gets straight in, and it's actually produces less issues with mitochondrial damage. The actual amount of macronutrients that in today's modern diet is probably balanced and geared more towards carbohydrate than it is towards fat. And when you've actually using fat in a higher proportion, then you actually maintain better the integrity of the actual mitochondrial membrane. So oxidative stress is something that we can actually look at within the laboratory through various means, which I'll discuss. And there's a list of oxidative stress in many chronic diseases: metabolic syndrome, diabetes, Alzheimer's, inflammatory bowel disease, pancreatitis, Parkinson's, which I believe will be discussed later. So one of the actual important enzymes that we can measure, which is critical to dealing with superoxide radical, is superoxide dismutase. This catalyzes the dismutation of superoxide into oxygen and hydrogen peroxide, and there's another enzyme that breaks that down. And so an early indication of increased production of superoxide dismutase is a good indication of oxidative stress. And then in addition to that, if you have deficiencies of copper, zinc, manganese, this will interfere with the actual activity in the production of the enzyme. So you're going to run into more problems there. So high levels of red cell superoxide dismutase suggest induction of the enzyme synthesis in response to oxidative stress. And we can measure this quite easily within the laboratory and provide information. Low levels of red cell superoxide dismutase suggest intracellular deficiencies of the enzyme or its cofactors, copper, zinc, and manganese. Another enzyme that's actually we can measure that's useful in determining oxidative stress is glutathione peroxidase. That's a tripeptide consisting of glutamic acid, cysteine, and glycine, and it's a substrate for glutathione peroxidase, which protects the cytosolic organelles from the damage of hydrogen peroxides formed by normal aerobic metabolism. It catalyzes the reduction of hydrogen peroxide to water and the simultaneous conversion of reduced glutathione to oxidized glutathione. So within the cells, you need to maintain a balance to be able to detoxify. And that's important. That's a super important fact as well. So This contains one residual molecule of cysteine, an analog of cysteine, in which selenium is substituted for sulfur. And so therefore, if you've got a deficiency of selenium, this will greatly decrease the activity of this enzyme. 8-hydroxydeoxyguanosine is a measurement. We can actually do this in urine, and it gives you a quantitative assessment of ongoing oxidative damage or stress in the body. It's an excellent biomarker of oxidative stress and a risk factor for various diseases including cancer. So excessive levels are associated with oxidative damage to lipids, proteins, and DNA. And this, the reductive oxygen species induced damage to nuclear and mitochondrial DNA occurs readily at the guanosine bases that are removed by DNA repair mechanisms. And these are actually excreted in the urine, and this is what we can actually measure. And as you can see, bladder and prostate cancer have been associated with elevated levels as well. So what are the nutrients for optimal function? And which ones do we regularly get requests for, and which ones can we get the most information from? So these are coenzyme Q10, carnitine, magnesium, vitamin B3, and iron. Carnitine, as many of you probably know, comes in two forms. You can have just standard L-carnitine, or you can have acetyl-carnitine. Generally, the standard L-carnitine is fine, but if you actually want to provide improved cognitive function, then you probably use acetyl-carnitine, because there's good evidence to suggest it actually crosses the blood-brain barrier. So coenzyme Q10. This has been an interest of mine for many years, because as part of my master's, I did a dissertation on coenzyme Q10, which actually was a long time ago, before it became popular to be put in toothpastes and creams and gels, and it was basically about the therapeutic uses of it, which was quite a long time ago. So now everyone has heard, most people have heard of it, and many people can gain support by using it, and that includes people with mitochondrial diseases, not just dysfunction, because there are many studies and papers showing that this can provide enormous support there. That just, I think, goes to show you the importance of it, because some of the genetic defects are, there is no cure. So coenzyme Q10 is a nutrient that can be synthesized by the body, but in amounts that are insufficient for metabolic needs. And so you need it in diet. It acts as a cofactor in electron transport, carrying electrons from complex 1 and complex 2 to complex 3. It's an important antioxidant, a vitally important antioxidant for membranes and the tissues where it's located. It's interesting in some part of my research that it's not really understood or known the exact dietary requirements. And I'm sure many of you will probably use maybe anywhere in the region of 50 milligrams to 100 milligrams a day. Some of the research I've come across suggesting up to 300 or even higher. So it's quite interesting. It's a variable, it's something that's actually obviously has a variability, probably based on biochemical variation, which varies from patient to patient. Everyone would have come across the difficulties that you're going to have if patients, clients are taking statins, because it, excuse me, statins inhibit the liver enzyme HMG-CoA reductase, to stop production of cholesterol, but they also have a detrimental effect on the production of CoQ10. So ideally, anyone that would be taking a statin should ideally be given support with CoQ10. So there are also other drugs that can block or interfere with the production. Beta blockers, diabetes drugs, tricyclic antidepressants. And it's a fact that actually CoQ10 will decline with age. So potentially people that, as you grow older, you're going to need more. That will vary depending on what you do in the meantime. I came across an interesting study recently that's going ahead in, it's a random controlled trial going ahead in Denmark. And they are looking to show that CoQ10 deficiency can possibly explain the long-term effects that develop with COVID-19 patients. Thanks. L-carnitine. So Rachel's already described that 60%, 70% of the energy actually is derived from fatty acids. And L-carnitine is actually a really important nutrient for the transportation of long-chain fatty acids into the mitochondria. The second function of carnitine is to regulate the intracellular mitochondrial ratio of coenzyme A to free coenzyme A. And this function is important because it allows the removal of excessive and potentially toxic short and medium chain fatty acids. So it's a vital nutrient. Red cell magnesium, Rachel did discuss this as well, and the importance with ATP. So after potassium, magnesium is the second most abundant cation within mammalian cells and the fourth most abundant cation in the extracellular water after sodium, potassium and calcium. 60%, this is quite an interesting fact actually, 60% of total magnesium is found in bone and 30% within muscle. So less than 1% of total body magnesium is actually in the extracellular space. So measurements of extracellular serum plasma magnesium are of limited use nutritionally, which is why we actually measure red cell magnesium. It has various important physiological functions. It's a cofactor for DNA and protein synthesis, for oxidative phosphorylation, for many enzymes. It's a cofactor for more than 300 different metabolic reactions, particularly those involved in energy use and storage. And so it's one of probably our most requested tests. And you'll understand why as you go through the day. Okay. In 1930, there was a Nobel Prize winner, biochemist, a Swedish biochemist, whose name was Hans Helsing. I don't think I pronounced that correctly, but anyway, he actually was dealing with alcoholic fermentation, and he won the Nobel Prize in chemistry for that. And he, in those days, this is 1930, he then knew that the importance of NAD was throughout every single animal and mammal and plants. So the actual importance, I think, of vitamin B3 is probably underestimated quite a lot. I know that if you go on Dr. Myhill's website, you'll see that she uses that for many patients with chronic fatigue. But I think it's as an importance throughout all cellular energy. It's one of the ones that we measure on a regular basis. So NAD, NADP carry hydrogen ions, protons from substrates through the mitochondrial electron transport chain. They're required for the function of more than 400 respiratory enzymes. Niacin requirements are thus based on energy expenditure. NADH and ADP are produced from the complete oxidation of glucose 6-phosphate. And then these reduced forms are then used in other metabolic processes such as fatty acid synthesis. So crucial. Iron. Iron is, as you'll know, iron is an important component of proteins involved in oxygen transport and metabolism, but it's also a heme functional form of iron synthesized by mitochondria. Heme is essential for hemoglobin, myoglobin, and deficiencies lead to reduced ability for electron transport chain complexes to function. So the important thing to note here is that before you consider supplementing with iron, you'd want to check the iron status, and ferritin is a good indicator of the overall body iron stores. Okay, so while we've been going through this exercise of reviewing some of the tests, we were also looking to try and achieve a new mitochondrial profile. And there's one or two tests that we are looking at now, and we are looking at trying to bring in on site to do this. And I'll quickly go through this in the last few minutes I've got left. The first one is alpha-lipoic acid. I'm sure some of you would have heard of it, maybe even supporting some of your patients' clients. It's a cofactor for mitochondrial enzymes in the final stages of glycolysis. Additional ALA can circulate in a free state where it has the ability to function as a water and a fat soluble antioxidant. So this is really quite unusual, and it's quite amazing if you think about it. You've got the water soluble antioxidants A, C, and E, and then you've got something like alpha-lipoic acid, which can support cell membrane health and cytosol as well. So it also has an important function in the role of production of glutathione. Now, this one you may or may have not heard of. I came across it quite a few years ago, but there's been an awful lot of research into it recently. So pyrroloquinoline quinone, PQQ, I think we'll just call it PQQ, protects from oxidative damage and stimulates the growth of new mitochondria. And this is where a lot of the research is actually coming at the moment from, the stimulation of new growth in mitochondria. Melatonin, we do get requests for melatonin, and it is important. And recently there's been a huge amount of research in the role of melatonin in mitochondrial health, because it's mainly synthesized by mitochondria, and it's a mitochondrial targeted antioxidant. And that's quite fascinating if you think about it. It protects the mitochondria from reactive oxygen species, inhibits the permeability pores, activates uncoupling proteins, which Rachel's already mentioned earlier, and therefore maintains optimal membrane potential and preserves mitochondrial function. So I think we're going to hear quite a lot in the coming years about melatonin. It also promotes mitophagy. Okay, thank you.
Thank you so much, Mark. That's very interesting. I imagine we've got time for a few questions. Any from the floor here? Dr. Burra. Oh, go ahead. Oh, we've got a mic.
Thank you so much, Mark. That was fantastic. I've got one quick question, one slightly longer question. With the measurement of superoxide dismutase, how much does the red cell superoxide dismutase that you measure reflect also mitochondrial or vascular salt activity or level of response that the patients can mount? So that's question number one. And then number two, I didn't realize mitochondria actually produce things like melatonin. And another thing you mentioned, so just a little bit more maybe on what else mitochondria make, the synthetic aspect of mitochondria.
Okay, I'll take the second one first then. I think as regards to melatonin, it's really quite a new area of
I
research.
think you may need to get nearer to the mic for the Zoom people.
It's good point. It's so in reference to melatonin, it's really quite an interesting new area of research, and I think there are several papers that I came across suggesting that melatonin actually gets produced even against a concentration gradient. So there's active transport within the mitochondria producing melatonin. There's got to be a real good reason for that. So this is a fascinating, for me, it's a fascinating area of new research. And I think going forward, you'll probably find that it's involved in a whole host of different aspects within mitochondria. But at this stage, it's probably too early to say. In terms of method, in terms of the assay for SOD, it's a really good overall indicator of oxidative stress. And this is where it becomes really difficult. And this is, I'm not sure if I mentioned earlier, but this is something where when you come to interpret the results of any test, it's got to be taken within the context of the patient. So we have many tests, as I've in the 30 minutes I've had, I've kind of whistled through quite a few. In relation to gaining information, ideally when you request a test, you really should go back to basics. You know, a test should answer a question. And therefore, if you've got a good clinical workup and you've got a profile or several different tests. So for example, if you did SOD, glutathione, magnesium, a couple of the other nutrients, and many of those were abnormal. And bearing in mind that all statistical tests are done following a Gaussian distribution, a normal distribution. So by definition, statistically, 95% plus minus 5% are going to be outside, low or high. So that is an important factor as well when trying to determine the actual clinical relevance of the result.
Oh, okay.
Just a very, very quick clinical comment. The overwhelming majority of the chronic fatigue in any patients I see are owls. You know, they don't get off to sleep until near midnight. They don't wake up. And to my mind, that's a pretty good biochemical test of melatonin deficiency.
Yeah. Yes, and circadian rhythm is I've spoken to many. So I've been with Biolab for about 2 years now. A hell of a 2 years it's been, but I come across quite regularly that there is often a missing component, if you like, for some of the issues that you see. And I think circadian rhythm is a critical component. And it's important, and it's quite, I wouldn't say simple to fix, but there are things that you can do to ensure that you have a good circadian rhythm. And the first thing is to limit the amount of blue light in the evening. And the second thing is to get up in the morning and get some natural light. And that will reset. It's like a reset, a reboot, if you like. And that is an important point. And obviously melatonin has function within that as well.
Good. Yes, as they say, the chair recognises Dr. Wendy Denning.
Thanks. This may not be the right person to ask this question to, but we are seeing increasing myositis post-COVID and post-viral infections. And my question, which is probably aimed at also practitioners, is how much of this do we think is a mitochondrial problem? How much of this do we think is an inflammatory problem? Because obviously they have similar but different approaches. What tests do we think are the most useful? And particularly in a patient, one of my patients who's got a raised creatinine kinase, for instance.
Right. Okay. So a raised CK, you know that there is muscle damage. So that's really you Did you know whether, did they do isoenzymes? No, so you just got a total CK. Was it grossly elevated?
It was double what it should be. But I've
That's not
seen that
grossly,
also with a patient.
that's
who's-
not grossly elevated.
I agree.
So it's mildly elevated. So what I see as grossly elevated is in cases of rhabdomyolysis, where you can have a CK of 10,000. It's a difficult question to answer, really. We obviously can measure inflammatory markers. Many of the markers I've been talking about are oxidative stress markers, but there are inflammatory markers, acute phase reactants such as ceruloplasmin, CRP, high sensitive CRP. It's an interesting question. I need to think about it.
Thank you. Yeah. If anybody else has any thoughts, I'd be grateful.
About
Question, and we're going to have to keep the time here. I need to put this one in first, please. Somebody on Zoom, I take it, is asking whether you can say anything about tryptophan as opposed to melatonin in this context. No.
It's a no, but perhaps they could expand on the question.
Okay. I'll
If
see
they can
if
expand on the question, and then
That's
they
fair
can
enough.
feed it through, and I will come back. Yeah. Yeah. Well, but in what, context?
Yeah. Okay, I'm not quite sure what they're asking, so if they get back to us, we'll see if we can pop it in later. Please.
Can I ask you about CoQ10 in terms of if it's not a drug-induced deficiency, what the likely causes of deficiency are, and which patients you'd be thinking we should be looking at their CoQ10 levels?
Okay, so CoQ10 is an interesting one because you're going to get it from a diet that's actually rich in certain foods. And all I can tell you is that vegetarian or vegan diets aren't. So basically, it's in good meats. And it's worthwhile thinking about the overall aspect of the diet, not just whether it's just vegetarian or vegan, because I understand that some of them will be good in nutrients and some will be supplementing. But CoQ10, I think, the more you age, the more it is difficult for the body to produce its own supply, and therefore diet becomes an even more a critical component. So, and it's also important, obviously, to measure it, change any dietary regimen or supplement, including supplement.
Thank you very much, Mark. I didn't know. I'm sorry, we've got to carry on that, really.